Thank you very much, and thank you all for hanging on till the bitter end. I know I'm the very last speaker, and usually when I get to the end of a meeting, I'm tired, my brain is full, so thank you for still opening your brains a little bit. And thank you also to the organizers for inviting me and for such a great meeting. I've organized meetings, so I know how much work goes into it, so really thank you very much. It's been a pleasure, and there's been a set of fantastic talks. Finally, thank you for the introduction. Those introductions always scare me. I feel like there's no way I can possibly live up to that, but here goes. So broadly speaking, what my lab is interested in is what we call the neural architecture of cognition. So I'd like to start by describing a little bit of what we mean by cognition, and we mean processes such as these. Working memory. How is it that you are able to keep some information in mind for a few seconds and then manipulate and use that mental representation maybe to drive a decision? Planning. How is it that you use knowledge of relationships between actions and their outcomes out there in the world to guide your decisions? For example, if you know that moving a pawn to a particular position will put pressure on a bishop, how does that inform your choice of what move to do in your next move in chess? What psychologists like to call executive or cognitive control. And I like to describe that as your capacity to control dynamically input-output relationships in your brain, to dynamically control how information gets routed within your brain. For example, I could say to you today, red means go and green means stop. And you could go out and you could drive that way, and it would take a little bit of mental effort, but you could do it. So how is it that just by deciding to pay attention to those instructions, you change red from going to the brake pedal to now red goes to the accelerator pedal? How is it that the brain does that? And also decision-making, evidence accumulation for decision-making. How is it that you accumulate evidence over time for different options, if you're making a choice across different options? And how do you use that accumulated evidence to eventually commit to a particular option and make the decision to follow that? These are the kinds of things that we study. And these are often studied with humans and often studied with fMRI and EEG and various other techniques. We're interested in trying to study them at the level of resolution of individual neurons and networks of individual neurons and networks across different regions of the brain. If you're interested in studying these processes at cellular resolution, you can do some work in humans like that, but it's difficult. Most of the work that is done studying these processes at cellular resolution is done with animals. And typically what we do is we train animals to perform behavioral tasks that we carefully design to try and isolate one or more of these processes and try and quantify the process very carefully. And what typically happens when you train animals in these kinds of tasks is that it takes a long time to train the animal in this very well-controlled task. The typical timescales that we encounter are months, and this is typical, I think, across all the field. And so that can be a very slow part. You have an idea, but it takes many months to train an animal, and then you're working on the idea, and so the cycle between an idea and testing it out in an experiment can be very, very slow. So we started trying to study these kinds of processes in rodents. I started doing this many years ago, more than 10 years ago. I was faculty at Cold Spring Harbor Labs, and my neighbors were Tony Zader and Zach Mainen, and they had started trying to use rats. At that moment, they were mostly using rats to study these kinds of processes, and I joined them in doing that. And our motivation is that rodents are small, they're relatively inexpensive, you can get good behavior out of them, and of course, they're amenable to modern molecular tools. And these two facts, that they're small and relatively inexpensive, mean that you can train many of them in parallel. So this picture is one of the walls in the behavior room in my lab where we train the animals. And here I'm showing you 9 out of the 36 behavioral rigs that we currently have. This is a computer rack, so a person is about that tall. And with 36 rigs, we train 9 rats per rig per day. We do 9 shifts per day. So we're training a total of about 324 individual rats every day. So through that, we get about 1 million behavioral trials a week. So that gives us a lot of statistical power in analyzing the behavior, and that's very nice. But also it means that we can train a lot of animals in parallel. So say a typical person in my lab supervises training of 30 to 40 animals, and say that you've dedicated 40 out of these 324 slots in a task that requires 4 months of training per individual animal. That means that if you're training 40 animals in parallel, on average, you get 1 trained animal out every 3 days. And that's wonderful compared to one trained animal every 6 months, right? That means you've got a lot more room to just try things out. Now, if you're going to train that many animals in parallel, there's no possible way that you can sit in front of each individual animal and shape it by hand and slowly see what it's doing and kind of like push it along in the behavioral shaping process. So the only way that you can train in this parallel mode is to write computer code that actually runs all the training, right? And that takes you through all the shaping steps, as in, oh, when I see that the animal did this, now respond this way, or provided a reward here, and that kind of stuff. That's what happens over all those months. And one thing that is very nice is that once you have that automated shaping code for the parallel training, it can take a long while to figure out what the code is. The animals often do things that you didn't expect they would do, and so you have to rewrite the code. So sometimes it's taken us years to get to write code for a particular task that works. But once you have that code, you can parallelize as much as you want. And so it's what makes this possible. And also, you can share that code with other people. Right? So we really like the idea of trying to use this to make the behavioral training more reproducible. Now, of course, there's many labs that are studying cognitive processes in rodents. Yusar spoke here, some wonderful work. For example, also studying cognitive control. Our particular interest and focus is in these complex type of tasks that I described in the first slide. I often refer to them as higher cognitive processes, but I put this in quotes because actually I don't really know what that means. I just know that it makes it sound kind of really cool and sophisticated. In practice, what it means is that we're working on tasks that takes us several months to train per individual animal, and so this whole system becomes really useful when you're kind of in that world. And using that system, that has really helped us push the envelope on what is known that rodents are capable of doing. So for each of the 4 cognitive processes that I mentioned in the first slide, we've been able to kind of push the envelope in terms of either showing that rodents are capable of performing these cognitive processes, or do them in a way that is particularly interesting, say, in comparison to how primates, including humans, do them. And so that's been really useful. It's really pushed us along, and it's really being driven by this large number of behavioral sessions per day, because as we develop these new behavioral tasks to study them in rodents, having many animals that you can test things out with really helps you in designing that task and developing the task. In comparison, when we come to trying to study each of these tasks, how it's implemented in the brain, which is really our interest, we've done a lot of work on behavior, but our interest really is how the brain implements these behaviors, things have been a lot more difficult. And part of the reason for that is that when we do, for example, electrophysiological recordings from the animals while they perform one of these tasks, well, the animals are wired. Most often, we've been using tetrodes so far, and so there are wires that come out and go to the amplifier, and our animals like to chew on the wires, so you need an experimenter there just to keep an eye on them and make sure that things go wrong. So because the electrophysiology is manned with individual people per session, really we've been running at about 3 to 4 electrophysiological or optogenetic sessions per day. So 2 orders of magnitude difference between those numbers, right? And so that has made our project of making the research much faster by parallelizing. It hasn't quite happened when we get to how things work in the brain. So we're trying to push on that as well, and we've been helped enormously by new wireless systems that have come out. There are various different flavors of wireless systems. We've been using some that were developed by Ala Karpova's lab at Janelia, and one of her postdocs, ex-postdocs, Matthias Carlson, now started a company called Spike Gadgets that makes these. And they're really great. So now we're doing wireless electrophysiological recording with 128 channels, and we're also doing wireless optogenetics. And for our particular case, these wireless systems marry really well with our behavioral system. And the thing that really makes them useful for us is that once they're wireless, we don't need a specialized rig for them. So we can run these in any one of our rigs. So what we're doing now is deploying them across our full behavioral rig. And so we're working towards soon having the same number of electrophysiological recording or optogenetic sessions per day as we have in behavior. And we hope that that will help us move a lot faster. So we've got these 4 different cognitive processes. Why are we studying these many different things in the lab? Well, it turns out that when you start to make guesses as to which brain regions might be involved in these different processes, you often start guessing about the same regions. Now, there's much that we don't know, so of course these are initial guesses and we're going to expand to different regions, but there's an overlapping set of regions that are the regions that you start thinking of when you think of any of these four. So we like having these four different types of projects in the lab because we think that they'll give us complementary information about these regions. And maybe by looking at them in these complementary ways, we'll be able to understand a lot better kind of what actually goes on with them. So today I'm going to tell you about two of these projects. And I've chosen them because they combine in this overlapping way. I'm going to tell you about a working memory project, and I'm going to tell you about an evidence accumulation for decision-making project. I'm going to start with this one, with evidence accumulation. And what I'm going to do is I'm going to start with perhaps the most common behavioral observation in decision-making. And this is something that is observed anecdotally. You observe it in your daily lives, which is that when you've got a decision that's very difficult to make, that can take a long time. But when you've got an easy decision, you make up your mind really fast. If you've received postdoc offers from two fabulous labs, you're going to think about it a lot. If you received a postdoc offer from a fabulous lab and a really stinky lab, you make up your mind really quick, right? So this has been observed also in the lab, not just anecdotally in your daily life. And it's been observed in all kinds of decisions, in social decisions, in sensory or perceptual decisions, economic decisions, gambling decisions, memory-based decisions, visual search decisions, value-based decisions, all kinds of decisions. It's been observed so commonly that it's thought that there's a common core process that sometimes goes under the name of noisy accumulation of evidence that it can account for this observation across all of these types of decisions. Some models that are used for this are sometimes called drift diffusion type models, so perhaps many of you have heard of those as well. So I'm guessing that many of you may know about these kinds of models, but let me take you through a caricature of them anyway for those of you that haven't. So the way it works is very simple, and it'll go like this. Pretend that you're a subject in an experiment in a lab where your task is going to be, you're going to be shown a sensory stimulus, and based on that, you're going to be asked to decide to either orient to the right or orient to the left. Okay? And now pretend that inside your brain, represented somehow, we don't know exactly how, that's going to be one of the questions that we want to address, there's a scalar variable that we're going to call the evidence accumulator A, and at the beginning of every trial, the value of that variable is 0. Okay? And now let's say that the trial starts, and you're observing the sensory stimulus, and every time that you see something that makes you lean towards going right, you add something to A, and every time that you see something that makes you lean towards going left, you subtract something from A. And you just keep going over time, adding and subtracting as you go, until you get to the end of the stimulus, the end of the trial, say maybe 2 seconds from the very start of the trial. And at that point, you have to make up your mind and report your decision. And in this model, what you do is simply ask, what's the sign of A? If A is bigger than 0, you're going to decide to go to the right. And if A is less than 0, you're going to decide to orient to the left. So A equals 0 is the decision board. Okay. Very simple model, but you can already see that it accounts for this observation. If the evidence is weak, then what you're adding or subtracting from A is going to be small. And that means that by the time you get to the end of the trial, maybe 2 whole seconds after the beginning of the trial, you're not going to be very far away from the decision border. If there's any noise in the process, not being far away from the decision border means that it's hard to tell which side you should lie on. So that's going to be a slow and a hard decision. Whereas if the evidence is strong and what you're adding or subtracting from A is very large, then you're going to move far away from the decision border very quickly, and that's going to become an easy and a fast decision. Okay? So super simple model. It's been around in the behavioral literature for about 50 years, and it turns out that it accounts for a ton of data across many, many different domains. Simple model that accounts for a lot of data. Of course, people love it, right? That doesn't mean that the model is right, but it's naturally very attractive. Now, in this model, the idea is that on any particular trial, the trajectory of this variable, the accumulator A, over time could be noisy. But if you average over similar classes of trials of similar difficulty, similar strength of evidence, you should see the trends that I described before. Namely, for weak evidence, the accumulator should grow only slowly, whereas for strong evidence, it should have a much steeper slope and should grow much more rapidly. Starting in 1996, Mike Shadlin and Bill Newsom and their colleagues started a series of experiments which were absolutely seminal in the field and started a whole school of studies And in these initial experiments, what they did was they recorded from monkeys in a particular part of the brain. They recorded from lateral and parietal cortex in monkeys. That's part of posterior parietal cortex. And in this talk, I'm just going to refer to it as posterior parietal cortex, or PPC for short. And they were recording from PPC while monkeys were forming a decision. They had to look at a stimulus and based on that stimulus decide whether to orient one way or the other. And they looked at the firing rates of the neurons that they were recording while the monkeys were forming the decision, before they had reported what the decision was. And what they saw there was that the firing rates of these neurons ramped over time, and for weak stimuli, for weak evidence, they ramped slowly, whereas for strong evidence, they ramped really quickly, exactly as had been predicted over here. So that was an extremely exciting moment, because that model had been around for decades, explained a ton of data, tied to a lot of behavioral experiments. And this was the first time that anybody had seen a signal inside the brain that actually correlated to that model, right? So super exciting things, suggesting that maybe indeed slow accumulation of evidence over time is a core part of how decisions get formed in the brain. Now, the similarity between these traces and these traces suggests that there's a one-to-one encoding between the firing rates of neurons in PPC and the value of the accumulator, right? There's a one-to-one map between, the value of the firing rates. You can ask what the value of the accumulator is, okay? And that was roughly the hypothesis that was proposed back then in 1996. And since then, there's been studies in many other regions, for example, in the frontal eye fields, another cortical region, where the same basic observation was made. For weak evidence, shallow ramping. For strong evidence, much faster ramping. So same idea, a one-to-one mapping between firing rates and value of the accumulator as it evolves over time. And these two cortical regions, PPC and FEF, are thought to be critical areas for this process of evidence accumulation. There's been much research over a long time in many labs, now extending to many other different regions. And here I'm showing you just 5 of the regions that have been studied. So other parts of prefrontal cortex, essentially the same idea. The striatum, the dorsal striatum, essentially the same idea. The superior colliculus in the midbrain, again, essentially the same idea. So a broadly distributed signal. And if you look at connections between these regions, and this little symbol means polysynaptic connection, not necessarily a monosynaptic connection. Of course, everything in the brain ends up being connected. Once you consider polysynaptic connections, everything is connected to each other. So they're all connected. And so this has led to the rough hypothesis that the similarity in the ramping firing rates that you see across different regions, maybe it means that the mechanism that leads to this accumulation process is a diffuse circuit mechanism, widely distributed across the brain. That could be. The way that I like to rephrase that is that we don't know what's going on. Right? You could look at the same data and say that. So when we started working on this problem, when we published our first paper on this 6 years ago now, these were some of the basic questions that were still out there that were still unanswered. Which of these brain regions is necessary or not necessary for the process wasn't known. Where is the evidence accumulation actually computed? Maybe it's none of these. Maybe it's somewhere else that hasn't yet been recorded from. That wasn't known. How is the evidence accumulation actually computed? That's what we would most like to know. Well, if you don't even know for sure that you've recorded from the region where it's happening, how do you know what the signals look like when the computation is actually being made, right? It's impossible to answer that question. So we thought, oh, we've got our rats. We can do this cycle between experiment and hypothesis and experiment a little faster. Maybe the rats will help us to unravel the mechanistic circuit logic underlying this. And we've always thought of this as a complementary approach to what is being done in the primates. Not at all kind of a replacement for the primates, but it's going to provide complementary information to what is being done with the primates. So the first question that we faced when we tried to do this is to ask, well, can rats actually gradually accumulate evidence for decision making over time scales that are similar to those that are seen in primates? Maybe rats just don't have this cognitive process, and it is absolutely pointless to try to study it in rodents, right? So to try and answer that, we came up with a test that we designed from the beginning for quantitative modeling to try and help us assess were the rats actually using this cognitive process or not. So let me take you through one trial in this task. And in this task, our animals face walls that have 3 nose ports in them. So nose ports are little conical openings that they can stick their nose into. And the way a trial starts is that we turn an LED on in the center port, and that's the signal to the trained animal, oh, you should put your nose into that center port, and you should keep your nose in there until we turn the light off. And we call that period the nose fixation period. And while the rat has its animal inside that center port, we play randomly timed auditory clicks from two speakers, one to their left and one to their right. Both streams of randomly timed clicks going at once. Click, 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 click. And then when those streams of clicks end, and the duration of the clicks varies randomly from trial to trial between 200 milliseconds up to 1.5 seconds. When those clicks end, we turn that center light off. That's the signal to the trained animal. Okay, now you can withdraw. And they should turn either to the right or to the left and put their nose into that side port. If they turn to the correct side, they get a water drop as a reward. We keep them thirsty. That's what motivates them to keep working. If they turn to the incorrect side, they get a white nose and a timeout, and then the next trial starts. So what defines which of the two sides is the correct side? It's which of the two sides, right or left, had played the greater total number of clicks. That's what tells them where the water reward is going to be. Okay. So why do we use that crazy form of artificial stimulus? Well, the reason is that if you were to perform this 100% correct, which nobody does, not humans, not monkeys, not rats, but if you were to perform this task 100% correct, then the only way that you could do it would be over time, add up the auditory clicks as they come in from the two sides, so that at the end of the trial, you can compare the two totals, right, and say which one had the greater total. Adding up auditory clicks over time, that is the same as accumulating sensory evidence over time, right? So we designed a task that we were trying to promote getting the rats to use a strategy of accumulation of evidence over time. So let me cartoon the processes that we think are important in this type of task. There's at least two very different periods in the task. First, while the clicks are playing, there's what we call the accumulation period. And you can roughly cartoon this as add one for each right click and subtract one for each left click from this accumulator variable. But then when you get to the end of the trial and that center light turns off, now what you have to do is compute what the sign of A is, and because the sign of A, that's what tells you either go to the left or go to the right. These are two very different computations, two very different periods, and that difference is going to become important a little while later in the talk, and I'll remind you about it when we get there. So one of the attractive things about using this kind of stimulus is that the click trains are Poisson, which means that they're as random as they could be. But we know exactly when we played each click to the microsecond, right? So that means that literally speaking, there are a lot of bits of information in each trial. And those bits of information, that gives us a lot of statistical power to allow us to estimate many parameters of the process. So that allowed us to quantify multiple parameters of this accumulation process. Some of those we were able to quantify for the first time. And it also gave us a lot of things to work with for analyzing our data to try and assess whether the rodents really were using gradual accumulation of evidence over time. We convinced ourselves that they were. I won't take you through the details of that. If you want to know the details, ask me when we come to the questions. But broadly speaking, there were three main things that pointed in that direction and that ended up convincing us. The first is we developed a relatively flexible model that could implement many different strategies for how the rats could solve this task. For example, one strategy might be, oh, whichever side has the greatest sudden burst of clicks, go to that side. And that will get greater than 50% correct, so maybe our rats were doing that. Right? So we had a model that, by varying the parameters of the model, it could implement this kind of burst detector or gradual accumulation of evidence over time, or many other different strategies or mixtures of those strategies. And then we fit the model to the data, and we asked, what do the parameters correspond to? And it turned out that the parameters corresponded to gradual accumulation over many hundreds of milliseconds over time, so that pointed to that strategy. Of course, that then leaves the question, well, how about using strategies that are not in the universe of strategies that your model can create, right? We tried to make a model that had many strategies in it, but still a finite universe. How about things outside it? So we've tried to come up with other ideas and other models or other strategies that we've been able to come up with and that we can quantify as a model, and we tried to fit to the data. None of them have fit the data as well as accumulation of evidence over time. So that way we've explored a little bit outside of the universe of the initial model. But then that still begs the question of like, Well, how about strategies that you haven't been able to think of just because you're not smart enough? Maybe the rats are using that. And so there what we've done is looked at predictions that our accumulation model makes, predictions of things that we didn't measure initially or that we didn't predict initially, and we've compared those to the data, and they're really well matched by the data. Okay? So all the current evidence right now points in that direction. There's still wriggle room, but we're at least convinced that it's a good working hypothesis. I want to point out that in the model that we developed, we have noise parameters in that model. So for any individual trial, so for any individual distribution of click times over the time of the trial, what the model evolves over time is not a deterministic trajectory of the accumulator variable, but a probability distribution of that variable and how it evolves over time. And then once we get to the decision point, if the animal on a particular trial actually turned to the right, that puts constraints on that probability distribution, which we can then propagate backwards. And so we're using all the behavioral information available, exactly what the stimulus was, exactly what the time of the clicks were, what is it that the rat responded, to constrain our estimate of how this accumulation variable evolved over time on each individual trial. And it turns out that having this estimate of the evolution of A over time for each individual trial and for each individual moment is really, really useful. And it's particularly useful when it comes to comparing to recordings that we do, neural recordings, while the animals perform the task. Because it means that for each individual trial, we have an estimate of how the accumulator variable evolved over time. And from any neuron that we record, we're also estimating the firing rate of that neuron and how that evolves over time. So that means that we can compare our estimate of the accumulator to the value of the firing rate at different time points and at different trials, and through that build a map of how the firing rate of the neuron depends on the value of the accumulator. In other words, we're building a tuning curve for the neurons that we record from. If you were recording from visual cortex and you're interested in how a neuron in visual cortex depends on the orientation of a bar that you're presenting in front of the animal, you want to plot firing rate as a function of orientation, firing rate as a function of the variable of interest. Well, people hadn't plotted these kinds of tuning curves for accumulated evidence because they didn't have this estimate of the accumulator over time, and if you don't have that estimate, you don't have anything on the horizontal axis with which you can plot, with respect to which you can plot. So having this estimate allowed us to estimate what we call these tuning curves, and those are going to be really useful when it comes to recording from different brain areas and trying to help us assess how do those brain areas represent this information. So having developed the behavior and having convinced ourselves that rats are using this strategy, we thought, okay, we can study it. Now let's go look for the regions that we think are involved in computing the evidence accumulation over time. So there were three characteristics that we thought that a region involved in this computation should have. The first is that the region should carry information about the graded value of the accumulator as it evolves over time. And these tuning curves are really going to help us to assess that. The second is that if we silence that region, that should impair the decision-making behavior. Right? If the decision behavior is based on accumulation of evidence and we've impaired a part of the brain that is involved in computing it, it should impair the behavior. Essentially, in practice, this translates into pharmacological perturbations. And also, remember that I told you that there are different periods in time? If a region is involved in computing the accumulation over this entire accumulation period, then if we silence or perturb that brain region at different points in time during the accumulation period, all of those different periods should affect the behavior. Okay? And so in practice, or methodologically, this is going to translate into optogenetic perturbations. So armed with these tools, now we're going to go out and hunt for where in the brain is this happening. So we're using rodents. Up to then, people had used primates to study this process. Where should we go? We have no idea. So what we'll do, or what we did at the time, was take some guesses based on the primate literature. Right? And I mentioned to you that these two regions, the PPC and in what rodents we call the FOF, the frontal orienting fields, which we think has a lot of similarities to the frontal eye fields of primates, that these two regions are thought to be particularly critical for the accumulation of evidence process. So there are two regions that seem to be similar in the rodents. We thought, let's start there. And so we started with those two, and I'm going to start by showing you data from the PPC. The first thing that we did was go record there while the animals are performing the task, and use our tuning curve tool to try and look at what these tuning curves look like. And what we found is that in the PPC, there's a smooth graded relationship between firing rates of neurons in the PPC and the value of the accumulator. This means that at any point in time during the trial, during that accumulation process, if you know the value of the firing rates in PPC, you can read off what the graded value of the accumulator is. Okay? Which in fact is exactly the hypothesis that they put forward back in 1996. So sometimes I like to describe this as we worked really hard to show that Mike Shadlin and Bill Neeson were correct in 1996. Of course, we did it in a different species and using different methods, and we think that there are advantages to this. But the essence of this picture is the same as what had been proposed back then. But then we did something that had not yet been done, which was simply silence PPC. Okay? And so here what I'm showing you is the percentage of trials in which the animal oriented to the right as a function of the total number of rights minus total number of left clicks. And in black, I'm showing you the control animals. And then in green, I'm showing you when we silenced the left PPC with mucimol, a GABA agonist. In red, I'm showing you when we silenced the right PPC. In blue, I'm showing you when we silenced bilaterally. And what you can see is that essentially nothing happened. So then we doubled the dose, and nothing happened. So then we quadrupled the dose, and nothing happened. So then we octupled the dose, and still nothing happened. So then we thought, you know, we're just not preparing our mucimol correctly. And so we introduced some control trials that were intermixed with the regular accumulation trials. And in those control trials, we saw a big effect. So we know that the mucimol is working. We know that we're silencing PPC. But this really tells us that silencing PPC has no effect on the accumulation of evidence behavior. So that rules out PPC as being central to the process. You know, maybe it's part of a bigger redundant circuit, and it plays a small role that is part of a bigger network of regions. But you know, these data tell us that it cannot be a central, essential region for it. Okay. So we'll come back to PPC later in the talk. And after these experiments, that inspired a monkey group, Alex Huck's group at the University of Texas at Austin, to do essentially the same experiments in monkeys. So they silenced PPC while they were performing the random dots task, and they found essentially the same results. And they used the same type of control trials, and again, they found an effect on the control trials, but not in the accumulation of evidence trials. So now we know that this result is not specific to rodents. It generalizes, at least to the monkeys. So we've ruled out PPC. Well, let's turn to the FOF. And the first thing I'll tell you about the FOF is that when we silence the FOF unilaterally, we see a big effect on the behavior. Okay? So basically, red and green curves are different from black. That doesn't tell us what role the FOF plays in this behavior, but at least it tells us that it plays some role. So that was nice to see. And then we recorded from the FOF while animals performed the task. And what I'm showing here in red is the tuning curve from the FOF. And in purple behind, I'm showing you the tuning curve that I'd shown you from the PPC. And what you can see is that the tuning curve from the FOF looks a lot more step-like. Okay? And so that means that if you know the firing rates of neurons in the FOF, you don't have a lot of information about the graded value of the accumulator, but you do have very good information about what the sign of the accumulator is. And that's because the edge of that step turns out to lie exactly at a equals 0. And I want to remind you, a equals 0 is the decision border. If a is bigger than 0, you should orient to the right. If a is less than 0, you should orient to the left. So that means that if you know the firing rates of the FOF at any point in time, you've got a very clear signal as to if the go signal came right now, which way should I go? Should I go left or should I go right? So that suggests that the FOF is actually involved in this process. It's part of the behavior, but it's more involved in this process, computing or representing the sign of A, than in this process. Now, you'll notice that I said, oh, the tuning curve looks step-like, but it's not a perfect step. And if you look near, this is an average over neurons. If you look over individual neurons, there are some neurons that have a very smooth representation. So while this suggested that the FOF might be more involved in this than this, it certainly doesn't nail it. So for that, we turned to optogenetics. We were using halorhodopsin, which allows us to silence when we turn the laser on after expressing halorhodopsin. We get silencing within about 16 milliseconds. When we turn the laser off, the activity comes back within about 60 milliseconds. So that means that we have high time resolution to probe which period in the trial is necessary. Is this region necessary? And so we did unilateral silencing of the FOF at different points in time during the trial. And so here in this plot, what I'm showing you, the y-axis represents the bias, the side bias that we induced. Again, it was unilateral silencing. And the extent of the horizontal bar tells you when we did the silencing. And so this bar tells you that when we silenced before the trial started, nothing happened. When we silenced during the first half of the accumulation period, nothing happened. When we silenced once the animals had started moving, nothing happened. But when we silenced during the second half of the accumulation period, that's when we saw an effect. That's when we saw an impairment of the behavior, an ipsilateral bias. And then we repeated the experiment, but now chopping time in half to look at higher time resolution. And we found essentially the same effect. Only when we silenced right near the time of this go signal was that when we saw an effect, as in FOF being necessary for the process, supporting the idea that the FOF is more involved in this than this. Okay? So that gave us some clarity as to what we think the FOF is doing. But it also meant that it isn't central to the gradual accumulation process, which is one of the central things that we're trying to study. Okay. So let's turn to other brain regions and see if we can find it. And at this point, we were a little worried as to how long it would take us to hunt through different places. And maybe it would take us 10 years to find a place that we were sure was involved. We also did experiments in the superior colliculus. I won't take you in detail through those. I'll just tell you that the results look very similar to those from the FOF. So we think that the superior colliculus is also more involved in this process than this process. And then Michael Yartsev, who is from Israel, he did his PhD at the Weizmann, and he was a postdoc in the lab. He said, let's go to the striatum. And I said, Michael, no, why? Why do you want to go to the striatum? It's going to be boring. And he said, no, I'm convinced I want to go. And I was like, OK, fine, because if you've met Michael, he's an energetic guy. He convinces you even when you don't want to be convinced, and he convinced me. And he was absolutely right to go there. So let me take you through that data. So he did recordings there, and we computed the tuning curve. And what we found in the anterior dorsal striatum, so is a subcortical region that receives connections from cortex. What we found is that there's a graded tuning curve, just like the PPC, in the sense that if you know the value of firing rates in the ADS, you can read off what the graded value of the accumulator is. But now, when we did the pharmacological experiments, or rather, Michael did them, when he did those pharmacological experiments, unlike the PPC, there was a big effect on the behavior. Okay? So we know that it's involved in this process in some way. And then he also did the optogenetic inactivations, and that's the data that I'm showing you here. For comparison, I'm showing you the FOF data in red. So once again, for the FOF data in red, it's only near the time of the go signal when you get an effect. But for the ADS, it didn't matter when during the accumulation period Michael turned on the silencing laser. He found an ipsilateral bias at any point in time during the accumulation period. So these are the three things that we set out to kind of like tick off as suggesting that a region is involved. Silencing affects the accumulation of evidence behavior. The firing rates in this region represent the graded value of the accumulating evidence as it evolves over time. And perturbing during any time point of the evidence accumulation period affects the behavior. Okay, so we're super excited about that because it suggests that finally we found a node in the brain that we think is causally involved in the process. Okay. The ADS receives projections from the FOF, and it's connected with the superior colliculus, which in turn feeds back to the FOF. So all these regions are interconnected. And we think that by studying these three regions, we'll be able to make a lot of progress in understanding the behavior as a whole. Because, of course, you not only want to study how the graded accumulation occurs, you also want to understand how the graded signal gets transformed into the binary signal that we see in the FOF and the SC to actually produce the go left versus go right command. And so what we're doing now, before I say that, I should point out that we don't think that the ADS is the only region necessary for the process. That might be. My guess is that that won't be true. But rather what's interesting to us is that by having found one node in the brain that we know is involved in the process, or we're fairly convinced is involved in the process, now we can follow the anatomy. It's a little bit as if we've grabbed onto the ankle of the thing that we were looking for, and now we can pull on the leg and get the rest. So what we're doing now is looking at brain regions that project to the ADS, or brain regions that this particular part of the ADS projects to try and unravel the whole circuit. And we're very excited about doing that. Also, we're recording simultaneously from these three regions. We're actually not doing all three at once. We're doing them in pairs, so the picture is a little bit misleading. And we're also doing simultaneous recordings of in one region and inactivations in another to try and understand how the regions interact with each other to produce this transformation from the graded signal into the binary categorization signal. So we're very excited thinking that we're at a kind of great moment in this research project, and that hopefully we'll finally unravel the circuit and actually understand how it works. Something that I haven't mentioned, everything that I've told you so far is analysis of single neurons, but we're also looking at population representations with multiply recorded neurons. So now I want to turn back to the other project that I mentioned, the working memory project. And the reason for that is that we left the PPC behind. I told you, oh, there are great signals. You can read out the accumulator there. Oh, but when we inactivate it, it doesn't do anything. So why does it have those signals? Plus, the PPC is strongly interconnected to the rest of the network. So this left behind a question of, like, what is it doing? And it turned out that this other project gave us some clues that applied in this one. So that's why I want to turn to this other project now. And to give you a sense of what the answer is, we think that the PPC is mediating, at least in the task that we're studying, long-term multi-trial effects of sensory history, their effects on the behavior. So let me take you through that data, and that's the work of an ex-postdoc, Athena Akrami, who now runs her own lab at the SWC in London. And she was using a different task, so big warning, the behavioral task is changing. I'm waving my hands to wake all of you up and make you notice that. It looks similar, but it's a different task. So in this task starts the same way with a center light that turns on, and the animals put their nose into that center port. And then we play an auditory sound that we call the first sound, SA. It's a pink noise, lasts 400 milliseconds. Then there's a delay that can last between 2 and 12 seconds. And then we play a second sound, SB. That's also a pink noise. And then after a short delay, a little go sound, boop. And the animals now have to, again, turn to one side. And the side that they should turn to, where the reward will be, is determined by which of these two sounds was the louder. Okay? And we call this a parametric working memory task because in order to solve this task, you have to remember how loud this one was throughout this delay. That's the working memory effect. In order to compare it to this one and be able to make this decision. And because loudness is an analog parameter, it's a continuous parameter, that's why we call it a parametric working memory task. And it's very convenient because it gives us something to plot against, right? So do this kind of working memory task is extremely useful. It's also identical, essentially, to the task that Merav Ahisar was talking about a couple of days ago. Was it yesterday or 2 days ago? I can't remember. But it's essentially the same task. And we see a lot of the same effects. And in fact, the sensory history effect that we see on behavior, that is the contraction bias that she and Jonathan have worked on. And in fact, a lot of the things that we did were inspired by their work, and as I'll tell you about in a second. So in this task, on any one trial, you choose a value of SA and a value of SB. So any one trial is defined in a two-dimensional plane. And the line SB equals SA, that defines the division between trials in which the animal should go to the left or trials in which the animal should go to the right. Okay? On any one trial, we choose a particular value of SA and SB, and over different trials, we present different pairs of values. This is the set of values that we've used, and the small number tells you the % correct that the animals were doing for that particular set of values. And we also use another set of values closely spaced around here because this is going to take us through different difficulties. How far you are from this diagonal line, that tells you how difficult each trial is going to be. If you're very far away from it, the loudness of one is very different from the loudness of the other. If you're on the line, they're both equally loud, and of course, it's impossible to say which was the louder. And so by using this closely spaced set of trials, that allows us to plot responses as a function of difficulty, and this is what responses for this particular set of trials look like. So if we plot percentage of trials in which the animals go to the right as a function of SA minus SB, we get these curves for these 3 example rats. And I'm going to compare that to the ideal performer. The ideal performer, every time that SA is louder than SB, should always go to the right, and every time that SA is softer than SB, it should always go to the left. Okay. Now, one thing that I haven't told you yet is that these data are from trials in which we were not turning a silencing laser on. So which silencing laser am I talking about? It turns out that we were interested in what the potential role of PPC might be in this task, because in the literature, and especially in the rodent literature, PPC is thought to be particularly important for working memory. This is a working memory task. We thought PPC is going to play a role. If we perturb PPC, in particular, if we silence it, we should impair behavior in these animals. So we expressed PPC bilaterally. We expressed halorhodopsin bilaterally in the PPC and put in 2 optical fibers. And so we were doing bilateral silencing. And we were intermixing trials in which the silencing laser was turned on with trials in which the silencing laser was off. These are the trials in which the silencing laser was off. So let me show you what happened in the intermixed trials when we turned the silencing laser on. And the big surprise to us is that on those trials, behavior looked closer to the ideal performer. So what we were expecting was that we would impair the behavior, and instead, the behavior got better. So that was not at all what we expected. So Athena did it in more rats just to see, is this really true? But yes, it was. She also did some recordings. So let me show you some of the data from those recordings. So here I'm showing you data from a typical PPC single neuron, and the different colored traces represent different values of the loudness of the first stimulus. And just to remind you, you have to remember that loudness during this delay interval so as to compare it to this other loudness and know where to go. And what you can see is that there's essentially no difference in firing rates across the different values of loudness of SA. In other words, the firing rates of PPC in the delay interval do not carry information as to the value of SA. There's no working memory neural correlate, at least in the firing rates here. Kind of disappointing, not what we expected. But then Athena kept recording during the intertrial interval. And what she found there was really surprising. What she found was that after the trial had finished, so this is when the go signal happens, the animals move around here, they've received their reward around here, they've moved back from that side port around here, and now they're just hanging out, waiting for the next trial. So once this information was completely useless for the current trial, that's when she saw this very clear separation in the firing rate, so very clear information about SA, about a trial that was already over. So that suggested to us, maybe we should be thinking about effects from previous trials in the past. And so that led us to work from here, from Rav Ahisar and Jonathan. And so inspired by them, we built a simple model of the behavior that takes into account trials in the past. Let me take you through that. And so in this simple model, we said, let's start with a model just for the current trial. We know the parameter value of how loud SA was and how loud SP was. Let's multiply each of those by a parameter, add them up with a bias parameter, put that through a logistic, and that should represent the probability that on that trial the animal went to the right, and we can fit that model to the data. And the magnitude of these parameters tells us, is a measure of how sensitive the behavior is to the stimuli that we're applying. We can do this for the current trial. We can also add in parameters for the previous trial's reward, for which side the animal was on the previous trial, for the previous trial's sensory stimulus, for the sensory stimulus 2 trials back, and for sensory stimuli even further back in time. Right? So this is very similar. So this is exactly what was inspired by these guys. And again, by looking at the magnitude of these weights, those are measures of how sensitive the behavior is to these parameters. And what we found is that the sensory history weights from one trial back and further back, they were significantly different from 0, smaller than the current trial parameters or other trial parameters, but significantly different from 0. And the thing is, we can fit this model to data from when the inactivation laser is on, or, and by the way, I should, just as a sanity check, of course, the model fits the behavioral data well. That just means that the fitting process worked. We can also fit them to trials where the inactivation laser was on. And the surprising thing that we found there is that when we did that, the sensory history weights, they all got smaller. But all the other weights were not significantly different from their control values. So quantitatively speaking, this was as if turning the inactivation laser on made the animal less sensitive to its sensory history. And in fact, what we can do is now again make a model prediction for what the behavior should look like by using these smaller values of the sensory history weights, but keeping the control values of these parameters. And when we do that, we get exactly a match to the improvement in the data that we'd seen, the improvement in the behavior that we'd seen in the rats. So quantitatively speaking, inactivating PPC eliminates the sensory history effects on the behavior. So I told you that the PPC represents sensory history. I showed you that in the firing rates. I also just told you that it mediates the effects of sensory history and behavior. Why is that interesting? Well, in part, all the links to what Mirav talked about, but also because there's a very large literature on sensory history effects. All the literature of Bayesian perception is about using a prior that's built up from your sensory history and how that affects your current performance. And there are very few circuit mechanisms that are hypothesized for that. Now, having found the PPC, this is similar to having found the ADS for the accumulation of evidence. We've got one foothold. We think that we can then pull on that and unravel the rest of the circuit, or at least that's what we hope to do. It also gave us a clue as to what might be going on in the first task that I talked about, that accumulation of evidence task. When we first did our experiments here, we weren't thinking of sensory history, so we didn't analyze sensory history. But now, after seeing this, we could go back and do that, and we found that in the PPC, in this task, once again, you find that the firing rates in PPC depend on the sensory stimuli of the previous trial, and we also found that in this task, silencing the PPC reduces the effect of sensory history on the behavior. So a consistent story across two very, very different tasks. I'm going to end there. I'm just going to make some concluding remarks. I'm first going to mention that almost everything I've told you so far has been about different brain regions and how they interact with behavior. We're not only interested at that level, we're also interested in going at a more finer grained level, at the level of individual neurons, and in thinking about different projection-defined and genetically defined neurons. And for that, we're starting to use more and more rat lines and working with mice. And I mentioned the high-throughput electrophysiology and optogenetics that we hope will help us move faster with this process. But what we're really trying to do is not just build a picture of how different brain regions correspond to parameters of the behavior, but how these regions interact with each other and how these different levels of analysis interact with each other to understand how the whole circuit leads to a particular behavior. And finally, just to thank the people that did the work, Athena Krami did all the parametric working memory work that I told you about. And these are the people that worked on the CLICS accumulation of evidence task. Bing developed the behavior. Jeff did pharmacology. Tim recorded from PPC and the FOF. Michael, as I mentioned, did the striatum work. Alex did some modeling work. And these are people currently in the lab pushing the project right now. And finally, thank you all for listening.